Hello students, welcome to the another video session of connective tissue. Since last three sessions we are trying to get get information about the connective tissue, and in last lecture we have seen about the skeletal connective tissue, and in this lecture, we are trying to get touch with the fluid connective tissue. This is the third main type of the connective tissue after the connective tissue proper. Skeletal connective tissue, the second, and third one is the fluid connective tissue. Mainly in animals, there are three types of fluid connective tissue are present: the blood, the tissue fluid, that is the our cytoplasm, and third one is the lymph or the hemolymph, which is present in the insects. So we are now concentrating on the first one, that is the blood. Blood consists of the mainly two types, that are the blood corpuscles, that are the free floating cells, and the second one is the uh, plasma, that is the fluid in the intracellular matrix, which is present with the these corpuscles, which provides the easily passage to float in the blood vessels. So in the first upper picture, you are seeing that the various types of the blood connective tissue we are going to see over there, that are the neutrophil erythrocytes, that means the red blood cells, and monocytes, lymphocytes, basophils, and eosinophils, and the platelets. These are the type of the uh, mm, blood cells, which WBCs. In the next picture, you are going to see uh, the slide of the blood smear, and this is a slide which stain with the one specific vital stain, and these all the gathering of these cells, which are in somewhat brick red in color, are the RBCs, and the spotted blue color are the platelets, or the platelets as well as the WBCs. So in an adult human body consists of 5 to 6 liters of blood in a total quantity of the of blood about the total 8% of his weight, the person's weight. Suppose a person is there and his weight is 55 kilograms, and uh, approximately 8% of the 55 kilograms are the 4.5 kilogram, and we know that the 1 kilogram is equal to 1 liter. From that formula, the person's weight is 4 point blood volume is 4.5 liters approximately. Okay, then we are moving towards the components of the blood. As you see in these picture, that the centrifuged one test tube, taking a sample of blood from one person, and it gets centrifuged, and the erythrocytes, the buffy coat, that are the leukocytes and platelets, are settled down uh, by the centrifugal force, and these are they settled down because their molecular weight, uh, um, they are heavier than the plasma, and plasma is uh, on the upper side. So there is 55% of the total blood is of plasma is of 55% of total blood. The buffy coat, that means the leukocytes and the platelets, are the less than 1%, but these less than 1% of the buffy coat is representing our total immune system, and it plays a very important role in our body. We will see it in next few slides. The erythrocytes are the 45% of total blood. and erythrocytes are nothing but the red blood corpuscles or the red blood cells. As we know that the liquid component of blood is about 90% water, and the remaining 10% of ions are proteins, nutrients, waste, and the dissolved gases. As we nutrients such as the amino acids, proteins, whether targeted proteins are there, or the proteins required for as in the form of nutrients to the muscles are also dissolved in the as a form of a food to the uh, particular cells. And the waste is generated in the particular cell is also get transported by the blood itself, and it get dissolved into the uh, blood, and then blood is get filtered with the kidney, and in the kidney, and our kidney filters these filtrate and make and these waste into the urine. Okay, and the dissolved gases such as the as we know that the dissolved gases, the blood supplies the oxygen to the basic unit of the body, that is the cell. Blood is a very important uh, connective tissue which is necessary for the easily ongoing of the humankind. The main protein in human plasma is albumin. Albumin is one specific type of protein which is present in the blood plasma. Some of the molecules found in the plasma with a specialized function such as the hormones, antibodies, and clotting factors. Clotting factors, hormones, so some targeted hormones which are secreted by the endocrine glands, and these targeted hormones are directly get into the blood and traveled up to the their targeted organ, and they stimulate that organ in by means of traveling through the blood. Antibodies, some pathogens or the foreign particles are entered into the our bloodstream, and they, as in last last or that last lecture, we have seen that the phagocytosis is done in the blood by the macrophages. The how that macrophages are going to know that the antibodies, the specific antibodies for the specific particle are there, so they are knowing, and these phagocytosis or the uh, they are alerting the immune system into the blood that the antibody plays a very important role over there. And blood clotting factor, if we have any blood clotting factor such as a thrombogen, and the thrombogenesis is occur over there, and the blood clot is generated, and the leaked blood vessel get repaired over there. So lipids such as cholesterol also present in the blood, and cholesterol is escorted with the some protein. Escorted means with cholesterol is attached with some protein to get traveled with the in the blood flow, because we know that the in blood 90% water is there, and the we know that the cholesterol is an fat, and fat is a hydrophobic in nature. That's why it can't be directly travel through the blood. That's why it needed to escort it from one to travel one place to another place. That why the cholesterol do not dissolve in water. So next part, that are the erythrocytes or the red blood corpuscles or the red blood cells. We are focusing on the red blood cells. The red blood cells are the specialized cells circulating through the body, and it delivers oxygen by the help of the hemoglobin. Small and biconcave, 7 to 8 micrometer in diameter, and they do not contain mitochondria or nucleus when they get mature. So why these RBCs are not contain any nucleus or mitochondria? That we know that the mitochondria is a powerhouse of the cell. Okay, but why these cells do not need the mitochondria? The hemoglobin have the four active sites on his uh, the molecule. Hemoglobin is a molecule which have the four binding sites, two for the carbon dioxide and two for the oxygen to transport. And these hemoglobin are in abundant number. It have to be in a large number in the each erythrocytes. That's why the some modifications are occur when the erythrocytes are generated by means of the hematopoiesis. And these erythrocytes getting some modification in it, that their degeneration of the nuclei and the nuclei and the mitochondria, which are the biggest cells, and they in need of the large size. That's why these modifications are occur in the evolution, that the nucleus and the mitochondria is get degenerated. That's why the space become more for the hemoglobin molecule, and more oxygen get transported through its targeted organ or the targeted cell. So the hemoglobin is a key protein used in oxygen transport. The red blood cells having the average life of the 120 days, and these red blood cells are degenerated after the or the phagocytosis is occur after the 120 days of these dead red blood cells, and get degenerated into the by the in the phagosome. The production of red blood corpuscles is controlled by the hormone erythropoietin, which is released by the kidneys. So kidneys plays very important role over here. They kidneys are also known as the one of type of the endocrine glands, and it also performs the filtration of the uh, by the of blood. And this erythropoietin is secreted by the kidneys in endocrine way, and this is get targeted through the red bone marrow, where the uh, red blood corpuscles are um, made of. Okay, so in this picture you are going to see the red blood cells flowing through the one artery, and these are the red in color. That's why this is the artery, and it's traveling or getting the RBCs, which are oxygenated, and this cross section of red blood corpuscles you are going to see over there, that the hemoglobin molecules are present in the abundance number in it. Okay, and that's the picture in below, that is the blood smear, and these stained or highlighted these big cells are nothing but the erythrocytes or the RBC or the red blood corpuscles. Let's move to the next uh, second part of the fluid connective tissue. First one we have seen the erythrocytes, and the next one is the leukocytes. That is nothing but the white blood corpuscles or the WBCs. And these leukocytes get divided into two parts, that are the granulocytes, and second part is agranulocytes. Let's focus what are the granulocytes. Granulocytes are nothing but the specific type of granules present in their cytoplasm. You are seeing that this picture that the of the granulocytes, that the blue dots are seen in these uh, the each cell, that are nothing but the granules present in the that cell in their cytoplasm. Three types of granulocytes mainly seen the cell name depending on the affinity for the neutral pH or acidic pH or the basic pH, or we can say the acidic dyes, neutral dyes, or the basic dyes. Hence they are named as the neutrophil. Neutrophil has the affinity to the neutral pH of the granules, which are the polymorphonuclear. Polymorphonuclear means what? The nucleus is in more than one nucleus is present in these neutrophils, as you can see, and these more than one number of the nucleus is connected by means of a thread like structure, and it performs uh, synchronized functions. Okay, and these neutrophils are polymorphonuclear. That's why it's known as the polymorphonuclear, the presence of the more than one nucleus. Okay, and its size varies from the 9 to 10 micron, and these neutrophils are present in the abundance number in the total number of WBCs, that are the 60 to 70% of total WBCs, and they are phagocytic in function. We have seen the process of the phagocytosis, and these neutrophils are the phagocytic in function. They are engulfing the foreign particles, and they are play in very important role in the our immune system. The next one is eosinophils. The eosinophils are the have affinity towards the acidic dyes, and these are the hence they are known as eosinophils. And the acidic granules are present in these eosinophils. These are the 2 to 4% in total WBCs presence in the blood, in the fluid connective tissue, that are the granulocytes of the total WBCs, 2 to 4%, and somewhat larger than the neutrophils. Their size is larger than the neutrophils, that is the 10 to 14 micron. They increase generally in a allergic condition. We have seen the allergic condition. We have seen the one particular drug act on these mucous cells, that are the cetirizine. We have seen its action and drug action over there, and these are released in the eosinophils are released in the allergic condition. The next third is the basophils. The basophils abundance in the total WBC are the 0.5 to 1%, and if you see their shape size, they are smaller than the neutrophils, and size varies from the 8 to 10 micron, and nucleus is large and irregular in shape. But one more thing in the eosinophil, I forgot to tell you that the you if you tell that the locate in the blood smear, that are the which type of cells are the eosinophils, and which type of cells are the neutrophils, or which type of cells are the basophils. You should focus on its nucleus. Okay, if you going to see the headphone type of a nucleus, then you will recognize that, or you should say that are the eosinophils, as the his nucleus of that cell is appear like the headphones, and multinucleated and connected by the thread are the neutrophils, and the H type of the its nucleus, and the larger among all these cells are the basophils. So these are the key factor or key points to identify the granulocyte, if someone tells you to identify. Let's focus on the basophils, and these are the 0.5 to 1%, and of total WBC, smaller than the neutrophil size, varies from the 8 to 10 micron, and nucleus is large and irregular in shape, and cytoplasm contain basophilic granules, as we say. That's why is known as the basophilic leukocytes. Okay, if you get the the key, that are the in number of the quantity, that are the neutrophils have the larger number than the eosinophils, and then comes the basophils, and in the size, The eosinophils are the larger, and neutrophils are second one, and the basophils are the last one. So this is for the extra sake of knowledge. And let's move towards the granulocytes. The granulocytes are non-granular. That the main key identification key is the granules are not present in the this type of the cells, and the nucleus is very big than the total, almost touching to the cell membrane. Okay, and these non-granular mononuclear, that the nucleus is only present in the single number of nucleus is present over there. And the leukocytes forms two types. These granulocytes forms two types, that are the lymphocytes and the monocytes. Okay, the lymphocytes size varies from the 6 to 10 micron, normally constitute about the 20 to 25% of the total WBCs, or the white blood corpuscles, that are important in the mechanism of immunity. And these monocytes plays very important role in the immunity, that the these cells produces antitoxins and the antibodies, all specific type of antibodies for the specific type of the pathogens, or the specific type of a coding are generated from the monocytes. So it has a very important role in the immune system, that it produces antibodies and the antitoxins also. Antitoxins are the specific chemical substance which deneutralize the power of the toxin. And these the identification key for the lymphocytes are the horseshoe shaped nucleus, and very big nucleus is seen in these monocytes. As you see in the this picture of the granulocytes, the first one, the lymphocyte, is the you can see that the nucleus of the cell is of horseshoe shape. Horseshoe means the we generally elaborate, or we generally name in Marathi the khur. It's of the टापा, घोड्याच्या टापासारखं दिसतंय. ओके, the second one is a monocytes, and monocytes are the large cells, constitute 3 to 8% of the total WBC, and of the total leukocytes varies from the 12 to 15 micron. And in active conditions, the monocyte produces the numerous pseudopodia. These are the, if you see the pseudopodia means the false legs. Podia means leg, pseudo means false. Okay, the false legs, there means, what do you mean by these are the wandering cells, or these are the migratory cells. Okay, so that's why they, if any foreign particle or the enters, and these lymphocytes get pseudopodia, and it can migrate from one place to another place by means of this pseudopodia. And these cells produces in the lymphoid tissue. Okay, the only the agranulocytes, or the monocytes, sorry, only the monocytes is produced in the lymphoid tissue, rather than the remaining all blood cells are produced in the red bone marrow. That may be the WBC and other things, like the red blood cell, or the other WBCs, or the platelets also get produced in the red bone marrow. So we will going to take the general idea about the thrombocytes or the platelets. What are the platelets? Generally, in the viral load, or the in malaria, we come to know that in our medical report, we the doctor says that the platelet count has decreased. And what how does these platelets are very important in our uh, blood system. We are going to touch that thing or that fact. These platelets are minute, and these platelets are the colorless bodies from the or forms the 2 to 4 micron in length, that is the very small size rather than the other cells of the blood system, and it circulates within the blood with a lifespan about the 10 days. Of the single platelet have the lifespan about 10 days. And the platelet as an inflammatory cells, how the inflammation they occurs, the and they are means the alerting cells or the alarm cells of the immune system, or the if thrombopoiesis. Okay, the thrombopoiesis is the process where the if any artery get ruptured and get from the blood is oozing out from the that artery, that the thrombopoiesis is occur over there. And the platelet formation is known as the thrombopoiesis, but the blood clot or the fibrin clot is over there uh, for the stop for the blood flow. So these blood clot requires or these thrombopoiesis means the genesis of these platelets. How these thrombo the clotting factor or the fibrin clot is a different thing. We will get touch with this, but get focus on the thrombopoiesis. Thrombopoiesis is the generation or the reproduction of the uh, platelets. How these platelets are get produced, and it requires thrombopoietin. Thrombopoietin is the chemical signal or the hormone which is secreted by the one of the major endocrine gland is known as the liver. Okay, thrombopoietin secreted from the liver get act on the megakaryocytes, that are the myeloid stem cells, and occurs in the myeloid tissue. Okay, this thrombopoiesis occur in the myeloid tissue, and these myeloid stem cells after the getting touch of the thrombopoietin get converted into the megakaryocytes. The megakaryocytes means what? Their number of the size of these myeloid stem cells get increased in numerous times, a number of times, and these form the megakaryocytes. And these megakaryocytes get fragmented, and means the platelets get fragmented from the megakaryocytes, and these number of platelets are produced from the one megakaryocytes, and these parental cell of these megakaryocytes are the myeloid stem cells. And these megakaryocyte fragmented into the number of cells, and hence the platelet are produced. And these platelet are important in the fibrin clotting, as well as clotting factor, as well as the inflammatory cells also. So let's touch with the what are the clot and what are the clotting factors. If fibrin is known as the clotting factor, and these fibrin get clot over the fibrin are the fibril long cells, that are the fibers which if any vein or the any artery get ruptured and the blood is oozing out, and these fibrin are get touch with the get collaborate with the platelets, and they are over that the ruptured place they get clot, and the blood clot is also happen over there, and the blood flow for the blood ruptured blood flow gets stopped by the this fibrin clot. So I have told you in last lecture that the we are going to take some idea about the hematopoiesis, that we all know until now that the uh, production of the red blood cells or the WBCs is occur in the bone, and the it occurs, and this production is known as the hematopoiesis. But how hematopoiesis occurs, I will try to get touch you with that. And these so in multipotent hematopoietic stem cells are known as the hemocytoblast. Okay, and these cells get divided into the two daughter cells. Okay, and the first daughter cell again get divide, or it remain as a stem cell for the production of the next two cells, and the which another second daughter cells get divided into the two main type, that are the myeloid stem cells and the lymphoid stem cells. The myeloid stem cells get divided into another four daughter cells, that are the megakaryoblast, proerythroblast, myoblast, and the monoblast. Okay, and again the megakaryoblast, as I said, in the megakaryocytes are the get developed, or they get size is bigger, get size on from the megakaryoblast, and megakaryocytes are producing platelets, and these precursor of the erythrocyte, or the parental cell of these erythrocytes, are the reticulocytes, and these reticulocytes are the get converted from the proerythroblast. The proerythroblast are the precursor cell of the erythrocytes itself, and after the proerythroblast get converted to the erythrocytes, when that are in the stage of a reticulocyte, that the degeneration of the nucleus and the mitochondria get occur over there, and these changing of that shape of this cell by means of invagination, and these uh, get converted into the non-nucleated erythrocytes. Okay, then the third one is a myoblast, is originated from the myeloid stem cells, and these myoblasts are the precursor cells of the granulocytes, that are the basophils, neutrophils, and the eosinophils. These, and the fourth one, that are the monoblast, and monoblast are the precursor molecule or precursor cell of the monocyte, and these monocyte after get old, that are the converted into the macrophages, which do the phagocytosis activity in the bloodstream. Okay, then the second main type after the myeloid stem cells, that are the lymphoid stem cells in this right corner upper part, and the lymphoid stem cells get converted into the lymphoblast. Okay, and this lymphoblast is converted into the natural killer cell, or the shortly known as the NK cells of the immune system, and these are the large granular lymphocytes. Okay, and the second daughter cell is get converted into the small lymphocytes, and these small lymphocytes are get converted into the T lymphocytes, B lymphocytes, and these T lymphocytes and B lymphocytes in you are going to see all these hematopoiesis, or these all in a special paper that is known as the immunology, and these B lymphocytes get converted after into the plasma cells. So these are the general idea of the hematopoiesis, and we are going to get in completion of the connective tissue, and in next lecture we are going to see the next part, next type of the connective tissue, that are the muscle tissue, and after that the last part of this chapter, second chapter, that are the nervous tissue itself. So let's get stop here, and hope we will meet soon in the next lecture. Okay, thank you students. uh, Thank you.